Chapter 12 Mineral Nutrition. In this chapter, we will study about method to study the mineral requirement of plants, essential mineral elements, mechanism of absorption of elements, translocation of solutes, soil as a reservoir of essential elements, metabolism of nitrogen. Okay. The basic needs of all living organisms are essentially the same. They require macromolecules such as carbohydrate, proteins, and fats, and the water and the minerals for their growth and development. This chapter focuses mainly on inorganic plant nutrition, wherein you will study the method to identify the elements essential to growth and the development of plants and the criteria for the stabilizing the essentiality. You will also study the role of essential elements, the major deficiency symptoms, and the mechanism of absorption of these essential elements. This chapter also introduces you briefly to the significance and the mechanism of biological nitrogen fixation. Method to study the mineral requirement of plants. In 1860, Julius von Sachs, a prominent German botanist, demonstrated for the first time that plants could be grown to maturity in a defined nutrient solution in complete absence of soil, and this technique of growing plant in a nutrient solution is known as hydroponics. Since then, a number of improvised methods have been employed to try and determine the mineral nutrients essential for plants. The essence of all these methods involves the culture of plants in a soil-free, defined mineral solution. These methods require purified water and mineral nutrient salts. Can you explain why is this so essential? After a series of experiments in which the roots of plants were immersed in nutrient solution wherein an element was added, substituted or removed or given in varied concentration a mineral solution suitable for the plant growth was obtained. By this method, essential elements were identified and their deficiency symptoms discovered. Hydroponics have been successfully employed as a technique for the commercial production of vegetables such as tomato, seedless cucumber, and lettuce. It must be emphasized that the nutrient solution must be adequately aerated to obtain the optimum growth. What would happen if the solution were poorly aerated? Diagrammatic views of the hydroponic technique is given in the figure 12.1 and 12.2. Essential mineral elements. Most of the minerals present in the soil can enter the plant through roots. In fact, more than 60 elements of the 105 discovered so far are found in the different plants. Some plant species accumulate selenium and some others gold, while some plants growing near the nuclear test sites take up radioactive strontium. There are techniques that are able to detect the minerals even at very low concentration, that is 10 to the power minus 8 gram per milliliter. The question is whether all the diverse mineral elements present in plant, for example gold and selenium as mentioned above, are really necessary for plants. How do we decide what is necessary for plants or what is not? Criteria for essentiality. The criteria for essentiality of elements are given below. First, A. The element must be absolutely necessary for supporting normal growth and the reproduction. In the absence of element, the plants do not complete their life cycle or the set of the seeds. The requirement of element must be specific and not be replaceable by another element. In the other words, deficiency of any one element cannot be met by the supplying some other element. The element must be directly involved in the metabolism of plants. Based upon the above criteria, only a few elements have been found to be absolutely essential for plant growth and metabolism. These elements are further divided into two broad categories based on their quantitative requirements. First, macronutrients and the second, micronutrients. Macronutrients are generally present in plant tissues in the large amount, that is in excess of 10 millimole per kg of dry matter. The macronutrients include carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, potassium, calcium, and magnesium. Of these, carbon, hydrogen, and oxygen are mainly obtained from the CO2 and H2O, while the others are absorbed from the soil as mineral nutrition. Micronutrients or trace elements are needed in a very small amount, that is less than 10 millimole per kg of dry matter. These include iron, manganese, copper, molybdenum, zinc, boron, chlorine, and nickel. In addition to the 17 essential elements named above, there are some beneficial elements such as sodium, silicon, cobalt, and selenium. They are required by the higher plants. Essential elements can also be grouped into the four broad categories on the basis of their diverse functions, and these categories are: first, essential elements as component of biomolecules and hence structural elements of the cells. For example, carbon, hydrogen, oxygen, and nitrogen. Second, essential elements that are components of energy related chemical compounds in the plants. For example, magnesium in chlorophyll and phosphorus in ATP. Essential element that activate or inhibit enzyme. For example, magnesium 2 plus is an activator for both ribulose bisphosphate carboxylase, oxygenase, and phosphoenol pyruvate carboxylase, both of which are critical enzyme in photosynthetic carbon fixation. Zn2 plus is an activator of alcohol dehydrogenase and molybdenum of nitrogenase during the nitrogen metabolism. Can you name a few more elements that fall in this category? For this, you will need to recollect some of the biochemical pathway you have studied earlier. Some essential elements can alter the osmotic potential of the cell. Potassium plays an important role in the opening and closing of the stomata. You may recall the role of minerals as solutes in determining the water potential of cell. Role of macro and the micronutrients. Essential elements perform several functions and they participate in the various metabolic processes in the plants, such as permeability of the cell, permeability of the cell membrane, maintenance of the osmotic concentration of cell sap, electron transport system, buffering action, enzymatic activity, and act as a major constituent of the macromolecules and the coenzymes. Various forms and the functions of the essential nutrient elements are given below. First, nitrogen. This is the essential nutrient element required by the plant in a greatest amount. It is absorbed mainly as nitrate, that is NO3 minus, through some are also taken up as NO2 minus or NH4 plus. Nitrogen is required by all parts of plant, particularly the meristematic tissues and the metabolically active cells. Nitrogen is one of the major constituents of the protein, nucleic acid, vitamins, and the hormones. Phosphorus. Phosphorus is absorbed by the plants from the soil in the form of phosphate ions, that is either as H2PO4 minus or HPO4 minus. Phosphorus is a constituent of the cell membranes, certain proteins, all nucleic acid and nucleotides, and is required for all phosphorylation reactions. Potassium. It is absorbed as potassium ions, that is K plus. In plants, this is required in more abundant quantities in the meristematic tissues, buds, leaves, and the root tips. Potassium helps to maintain an ion-cation balance in the cells and is involved in the protein synthesis, opening and the closing of stomata, activation of enzymes, and in the maintenance of the turgidity of the cells. Calcium. Plant absorb calcium from the soil in the form of calcium ions, that is Ca2 plus. Calcium is required by the meristematic and differentiating tissues. During the cell division, it is used in the synthesis of cell wall, particularly as calcium pectate in the middle lamella. It is also needed during the formation of mitotic spindle. It accumulates in older leaves. It is involved in the normal functioning of the cell membrane. It activates the certain enzymes and plays an important role in regulating the metabolic activities. Now, magnesium. It is absorbed by the plants in the form of divalent, that is Mg2 plus, and it activates the enzyme of respiration, photosynthesis, and are involved in the synthesis of DNA and RNA. Magnesium is a constituent of the ring structure of chlorophyll and helps to maintain the ribosome structure. Sulfur. Plants obtain sulfur in the form of sulfate, that is SO4 minus. Sulfur is present in two amino acid, that is cysteine and the methionine, and is the main constituent of the several coenzymes, that is vitamins, that is thiamine, biotin, coenzyme A, and the ferrodexin. Iron. Plants obtain iron in the form of ferric ions, that is Fe3 plus, and it is required in the larger amount in the comparison to the other micronutrients. It is important constituent of the proteins involved in the transfer of electrons like ferrodexin and the cytochromes. It is reversibly oxidized from Fe2 plus to Fe3 plus during the electron transfer. It activates catalase enzyme and is essential for the formation of chlorophyll. Manganese. It is absorbed in the form of manganese ions, that is Mn2 plus. It activates many enzymes involved in the photosynthesis, respiration, and nitrogen metabolism. The best defined function of manganese is in the splitting of water to liberate oxygen during photosynthesis. Plants. Zinc. Plants obtain zinc as Zn2 plus ions. It activates various enzymes, especially carboxylases. It also needed in the synthesis of oxygen. Copper. It is absorbed as a cupric ions, that is Cu2 plus. It is essential for the overall metabolism in plants. Like iron, it is associated with the certain enzymes involved in the redox reaction and reversibly oxidized from Cu2 plus to Cu plus to Cu2 plus. Boron, it is absorbed as BO3 3 minus or B4O7 2 minus. Boron is required for uptake and utilization of calcium, membrane functioning, pollen germination, cell elongation, cell differentiation, and carbohydrate translocation. Molybdenum, plants obtain it in the form of molybdate ions, that is MoO22 plus. It is a component of several enzymes including nitrogenase and nitrate reductase, both of which participate in nitrogen metabolism. Chlorine, it is absorbed in the form of chloride ion, that is Cl minus, along with the sodium ion and potassium ion. It helps in the determining the solute concentration and anion-cation balance in the cells. It is essential for the water splitting reactions in the photosynthesis, a reaction that leads to the oxygen evolution. Deficiency symptom of the essential elements. Whenever the supply of essential elements become limited, plant growth is retarded. The concentration of the essential element below which the plant's growth is retarded is termed as critical concentration. The element is said to be deficient when present below the critical concentration. Since each element has one or more specific structural or the functional role in plants, in the absence of any particular elements, plants show certain morphological changes. These morphological changes are indicative of certain element deficiencies and are called deficiency symptoms. And the deficiency symptoms vary from element to element, and they disappear when the deficient mineral nutrient is provided to the plant. However, if deprivation continues, it may eventually lead to the death of the plant. And the parts of the plant that show deficiency symptom also depend on the mobility of the element in the plant. For elements that are actively mobilized within the plants and are exported to the young developing tissues, the deficiency symptom tend to appear first in the older leaves. For example, the deficiency symptom of nitrogen, potassium, and magnesium are visible first in the senescent leaves. In the older leaves, biomolecules containing these elements are broken down and making these elements available for mobilizing to younger leaves. The deficiency symptoms tend to appear first in the young leaves tissues whenever the elements are relatively immobile and are transported out of the mature organs. For example, elements like sulfur and calcium are a part of structural component of cell and hence are not easily released. This aspect of the mineral nutrients of plants is of a great significance and importance to agriculture and horticulture. The kind of deficiency symptoms shown in plants include chlorosis, necrosis, stunted plant growth, premature fall of leaves and buds, and the inhibition of cell division. Chlorosis is the loss of chlorophyll leading to the yellowing in the leaves. This symptom is caused by the deficiency of the elements like nitrogen, potassium, magnesium, sulfur, iron, manganese, zinc, and molybdenum. Likewise, necrosis or the death of tissue, particularly leaf tissue, is due to the deficiency of calcium, magnesium, copper, and potassium. Lack or the low level of nitrogen, potassium, sulfur, molybdenum causes inhibition of the cell division. Some elements like nitrogen, sulfur, molybdenum delay flowering if their concentration in the plants is low. You can see from the above that deficiency of any element can cause multiple symptoms, and the same symptoms may be caused by the deficiency of one of the several elements. Hence, to identify the deficient element, one has to study all the symptoms developed in all the various parts of the plant and compare them with the available standard tables. We must be aware that the different plants also respond differently to the deficiency of the same element. Toxicity of the micronutrients. The requirement of micronutrients is always in the low amount, while their moderate decrease causes the deficiency symptoms and a moderate increase causes toxicity. In other words, there is a narrow range of concentration at which the elements are optimum. Any mineral ion concentration in the tissues that reduces the dry weight of tissue by about 10% is considered toxic. Such critical concentrations vary widely among the different micronutrients. The toxicity symptoms are difficult to identify. Toxicity levels for any elements also vary for the different plants. Many a times, excess of the element may inhibit the uptake of another element. For example, the prominent study of the manganese toxicity is the appearance of the brown spot surrounded by a chlorotic veins. It is important to note that manganese competes with the iron and magnesium for the uptake, with the magnesium for binding with enzyme. Manganese also inhibits calcium translocation in the shoot apex. Therefore, excess of manganese may in fact induces deficiency of iron, magnesium, and calcium. Thus, what appears as a symptom of manganese toxicity may actually be the deficiency of symptoms of the iron, magnesium, and calcium. Can this knowledge be the sum important to a farmer, gardener, or even for your kitchen garden? Mechanism of absorption of elements. Much of studies on mechanism of absorption of elements by plants have been carried out in isolated cells, tissues, or organs, and these studies revealed that the process of absorption can be demarcated into the two main phases. In the first phase, an initial rapid uptake of ions into the free space or the outside space of the cells, that is the apoplast, is a passive. In the second phase, the ions are taken in slowly into the inner space, that is the simplest of the cells. The passive movement of ions into the apoplast usually occurs through the ion channels, the transmembrane proteins that function as a selective pores. On the other hand, the entry or the exit of ions to and from the simplest requires the expenditure of metabolic energy, which is an active process. The movement of ions is usually called the flux, and the inward movement into the cells is influx, and the outward movement is efflux. You have read the aspect of the mineral nutrient uptake and translocation in the chapter 11. Translocation of solutes. Mineral salts are translocated through xylem along with the ascending stream of the water, which is pulled up through the plant by transpirational pull. Analysis of xylem sap shows the presence of mineral salts in it. Use of radioisotopes of mineral elements also substantiate the view that they are transported through the xylem. You have already discussed the movement of water in the xylem. Soil as a reservoir of essential elements. Majority of the nutrients that are essential for growth and the development of plants become available to the roots due to the weathering and the breakdown of rocks. These processes enrich the soil with the dissolved ions and inorganic salts. Since they are derived from the rock minerals, their role in the plant nutrition is referred to as the mineral nutrition. Soil consists of a wide variety of substances. Soil not only supplies minerals but also harvests nitrogen fixing bacteria. Other microbes, holds water, supplies air to the roots, and act as a matrix that stabilizes the plant. Such deficiency of essential minerals affect the crop yield, and there is often a need for supplying them through fertilizers. Both macronutrients, that is nitrogen, phosphorus, potassium, sulfur, etc., and the micronutrient, that is copper, zinc, iron, manganese, etc., form the component of fertilizers and are applied as per need. Metabolism of nitrogen, that is nitrogen cycle. Apart from carbon, hydrogen, and oxygen, nitrogen is the most prevalent element in the living organism. Nitrogen is a constituent of the amino acid, proteins, hormones, chlorophylls, and many of the vitamins. Plants compete with the microbes for the limited nitrogen is available in soil. Thus, nitrogen is a limiting nutrient for the both natural and agricultural ecosystems. Nitrogen exists as two nitrogen atoms joined by a very strong triple bond, that is N triple bond N. The process of conversion of nitrogen to the ammonia is termed as nitrogen fixation. In nature, lightning and the ultraviolet radiation provide enough energy to convert nitrogen to the nitrogen oxide, that is NO, NO2, and N2O. Industrial combustion, forest fires, automobile exhaust, and the power generating station are also sources of the atmospheric nitrogen oxides. Decomposition of organic nitrogens of dead plants and animals into the ammonia is called ammonification. Some of the ammonia volatilizes and re-enters the atmosphere, but most of it is converted into nitrate by the soil bacteria in the following steps. 2 mol of NH3 plus 3 mol of oxygen form 2 mol of nitrate, NO2 minus, and 2 mol of H plus and 2 mol of water. And here 2 mol of NO2 minus plus 1 mol of oxygen, and it forms 2 mol of NO3 minus. Ammonia is first oxidized to nitrate by bacteria Nitrosomonas and or the Nitrococcus. The nitrite is further oxidized to nitrate with the help of the bacterium Nitrobacter. These steps are called nitrification, and the nitrifying bacteria are chemoautotrophs. The nitrate thus formed is absorbed by the plants and is transported to the leaves. In leaves, it is reduced to form ammonia that finally forms the amine group of amino acid. Nitrate present in the soil is also reduced to the nitrogen by the process of denitrification. Denitrification is carried by the bacteria Pseudomonas and the Thiobacillus. Biological nitrogen fixation. Very few living organisms, can utilize the nitrogen in the form of N2, Available evidently in the air, only certain prokaryotic species are capable of fixing nitrogen. Reduction of nitrogen to ammonia by living organism is called the biological nitrogen fixation. The enzyme nitrogenase, which is capable of nitrogen reduction, is present exclusively in the prokaryotes. Such microbes are called nitrogen fixers. N triple bond N in the presence of nitrogenase form the ammonia. The nitrogen fixing microbes can be free living or symbiotic. Example of the free living nitrogen fixing aerobic microbes are Azobacter and the Weissennichia, while the Rhodospirillum is anaerobic and free living. In addition, number of cyanobacteria such as Anabena and the Nostec are also free living nitrogen fixers. Symbiotic biological nitrogen fixation. A special type of symbiotic biological nitrogen fixation association are known. The most prominent among them is the legume bacteria relationship. A species of rod-shaped rhizobium has such relationship with the root of the several legumes such as alfalfa, sweet clover, sweet pea, lentils, garden pea, broad bean, clover beans. The most common association of the roots as nodules, and these nodules are the small outgrowth on the roots, and the microbes Frankia also produces nitrogen fixing nodules on the root of the non-leguminous plant, that is example alnus. Both rhizobium and the frankia are free living in soil but as symbiotic can fix atmospheric nitrogen. Uproot any one plant of a common pulse just before flowering. You will see near spherical outgrowth on the roots. These are nodules. If you cut through them, you will notice that central position is red and pink. What makes the nodules pink? And this is due to the presence of a leguminous hemoglobin or the leghemoglobin. Nodule formation. Nodule formation involves a sequence of multiple interaction between the rhizobium and the roots of the host plant. Principal stages in the nodule formation are summarized as follows. Rhizobium multiply and colonize the surrounding of roots and get attached to the epidermal cells and the root hair cells. The root hair curl and the bacteria invade the root hair. An infection thread is produced carrying the bacterium into the cortex of the root, where they initiate the nodule formation in the cortex of the root. Then the bacteria are released from the thread into the cells, which leads to the differentiation of specialized nitrogen fixing cells. The nodule thus formed establishes a direct vascular connection with the host for the exchange of nutrients. These events are depicted in the figure 12.1. The nodule contains all necessary biochemical components such as the enzyme nitrogenase and leghemoglobin. The enzyme nitrogenase is a molybdenum iron protein and catalyzes the conversion of atmospheric nitrogen to ammonium, and the first stable product of nitrogen fixation. The reaction is as follows. N2 plus 8 electron plus 8 H plus 16 ATP forms 2 mole of ammonia plus H2 plus 6 ADP plus 16 phosphate. The enzyme nitrogenase is highly sensitive to the molecular oxygen. It requires anaerobic condition. The nodule have adaptation to ensure that the enzyme is protected from oxygen. And to protect these enzymes, the nodule contains an oxygen scavenger called the leghemoglobin. It is interesting to note that these microbes live as aerobes under the free living condition where nitrogenase is not operational, but during the nitrogen fixation events they become anaerobic, thus protecting the nitrogenase enzyme. You must have been noticed in the above reaction that the ammonia synthesized by the nitrogen requirement. Very high input of energy, that is 8 ATP for the each ammonia produced. The energy required thus is obtained from the respiration of the host cells. Fate of ammonia. At physiological pH, the ammonia is protonated to form NH4+, that is ammonium ion. While the most of the plants can assimilate nitrate as well as ammonium ions, the latter is quite toxic and to plants and hence cannot be accumulated in them. Let us now see how NH4+ is used to synthesize amino acid in the plants. There are two main ways in which this can take place. Reductive amination. In these processes, ammonia react with the alpha ketogluteric acid and forms glutamic acid as indicated in the equation given below. Alpha ketogluteric acid plus ammonium ion plus NADPH in the presence of glutamate dehydrogenase, glutamate plus H2O plus NADP is formed. Transamination. It involves the transfer of amino group from the one amino acid to the keto group of a keto acid. Glutamic acid is the main amino acid from which the transfer of NH2, the amino group, takes place, and the other amino acid are formed through the transamination, and the enzyme transaminase catalyzes all such reactions. Amino donor, amino acceptor, and it forms. The two most important amides, that is asparagine and the glutamine found in the plants, are the structural part of proteins. They are found from the two amino acids, namely aspartic acid and the glutamic acid respectively, by the addition of another amino group to each. The hydroxyl part of the acid is replaced by the another NH2 radical, and since amides contain more nitrogen than the amino acid, they are transported to the other parts of the plant via xylem vessels. In addition, along with the transpiration stream in the nodules of some plants, for example soybean, export the fixed nitrogen as ureates. These compounds also have a particularly high nitrogen to carbon ratio.